Today we're going to be learning the streak plate technique. Okay, so we are going to be creating a streak plate. Streak plate technique is used to create a plate that allows you to have isolated colonies. So it'd be very useful for something that's like a mixed culture of bacteria, or say a swab that's a culture from a wound or something, and you're trying to isolate what kinds of bacteria are present. Or if you have a mixed culture and you only want one particular species out of it, or if you want to see what species are in it, this will allow you to create isolated colonies of each of the species so that you have a pure culture of each to work from in the future. So you will need your inoculating loop once again. Okay, and make sure your wire's straight. And this time it'll be kind of important to have that little crook on it. It's easier so that you don't dig into the agar. Remember, it is not a really solid media. It's a semi-solid media like a jello, so you can dig into it easily. Okay, so it lets you skate along without digging into it. We have a mixed culture of Staphylococcus epidermidis and Serratia marcescens. So there's two species of bacteria in this broth culture. Okay, remember when you agitate it to resuspend the cells to do it back and forth and not up and down. Okay, so this mixed culture, what we're going to do is create a streak plate that will allow us to separate those two species of bacteria from each other so that we have a pure culture of each on the plate. The way that's going to be done is to create isolated colonies of each of these bacteria. And an isolated colony is one cell created from one cell that has gotten spread apart from the rest of them, so that colony that you can actually see, visibly see, has been created from one cell. So it's a clone of that one cell, which makes it a pure culture of that one cell. So we want to get one cell of Staphylococcus epidermidis away from everything else so that it creates a colony of just Staphylococcus epidermidis, and it's a pure culture of that. And we want to get one cell of Serratia marcescens far enough away from all the other cells to create a colony of Serratia marcescens that was the result of one cell, so that it's a pure culture because it was created from one cell and clones of that one cell. Okay, so that is the culture we're using, and you will need a nutrient agar plate. Remember, we use plates lid side down. I have labeled this. Okay, you want to label it with both species of bacteria because both of them are going to be on here. Your date and the initials, your initials. Okay, so we also need a Bunsen burner, which we'll go ahead and light. Okay, I've put a handout on that goes through the background and the procedure. Okay, and on the second page there is a little diagram that helps you to follow what I'm doing. Okay, I'll do it in more detail. So I'm going to do it on the plate first, and it's kind of hard to see maybe on the video for the plate, but I will also do it on the board with a Sharpie and go over it. So we have our plate, we have it labeled, and we are going to sterilize our loop. Remember, we're using aseptic technique that we're really good at by now. Okay, so we let it turn orange. Remember, it's hot. You're going to pick up the culture you're trying to isolate the bacteria from. Remember, hold it by the glass. Take the lid off with your little finger. Flame the top. Remember, this is hot. Remember to point this away from your face. Go in, cool it on the inside of the tube, and then go in and get a loop full of bacteria. Okay, and then flame the top. Put the lid on and put that down. Now you will not go back to that again, because remember, you're trying to dilute this one loop full of bacteria. This is another dilution mechanism. Okay, so pick up your plate with your loop full of bacteria. You are going to streak the first third, okay, the top third of the plate, just back and forth gently. Don't cut into the agar, just skate along the agar. Okay, so you're done with your first streak at that point. Okay, and that streak is going to contain the most amount of bacteria because it came right from the tube. Okay, then you're going to sterilize your loop. Okay, now this time you do not go back to the bacteria, because if you did, you're not diluting the bacteria. What you want to do is dilute the bacteria that's already on the plate in the first streak. Okay, so remember, this is hot. What you do is turn your plate about a third clockwise, okay, and that way when you pick it up, the part where you want to put the second streak is conveniently at the top, making it easier. Okay, remember, this is hot. Make sure you cool it where you haven't put bacteria, and you'll be able to see. You can see a little bit of the scrape marks from putting it on the agar. Okay, so I'm going to cool it down here where there's no bacteria yet. And then to make my second streak, I'm going to go into that first streak a few times and create a second streak on that other third, the second third of the agar plate. Okay? And then you put it down. Remember to sterilize your loop. Okay? So there's no bacteria on this loop. Again, you do not go back to the original culture. Okay, you're diluting that one first streak. Turn your plate that last third clockwise. Okay, and this will be your third streak. You pick it up. Again, cool this loop where you haven't put any bacteria yet. Okay, and then from your second streak, you're going to go into that a few times and create a third streak. And kind of draw it out at the bottom if you can, if you have the room. That way you're dragging your cells further and further away from each other, so you're more likely to get isolated colonies. Okay, so then you put this back down, make sure you sterilize your loop. Okay, and you have now completed a streak plate. Okay, so I'm going to put it on the board now so that it might be more visible. So I'm going to shut this Bunsen burner off because we're not going to physically use it, but I'm going to pretend it's on, and I'm going to find a marker, maybe. Okay, so what we want to do is pretend this marker is our inoculate group. Okay, so what we're going to do is we've got our agar plate. Okay, so we are going to sterilize our loop. We're pretending this is our loop. We're going to sterilize our loop. We're going to pick up our culture, flame it, remove the lid, go in, get a loop full of bacteria, flame the top, put the lid back on, put it down. Take this loop full of bacteria to make your first streak. So you're going to streak across the first third of that agar plate, okay, that nutrient agar plate. Okay, so the first streak has been done now. Then you'll take your inoculating loop. You'll sterilize it again. Okay? You do not go back to the bacteria because you're trying to dilute this first streak, so you don't want to add any more bacteria. Okay, so remember this is still a little hot. Remember you cool it someplace that you haven't put the bacteria yet. Okay, now it's cool. Then you do your second streak by going into that first streak some of the time. That way you're dragging some of the cells into your second streak, but not a lot of them, just a few. Okay? Then you sterilize your loop again. Okay? And then you cool it again where there is not bacteria present yet. Then you're going to finish with your third streak. And this is the streak you should get isolated colonies in as you drag the cells further and further away from each other. So, do your third streak. You're going to go back and forth on that third streak, coming into the second streak a little bit, dragging some cells, and then kind of go out and trail along if you have some room so that you can try and keep dragging cells further and further out. So after incubation, okay, which would be 24 to 48 hours at 37 degrees Celsius. Okay, after incubation, hopefully you're going to have isolated colonies down in here, something like that, okay? Up here you should have a very solid streak of bacteria. That's where the most bacteria is because that's when you got it right out of the tube, okay? That is the most condensed part, has the most growth. Second streak, there should be a little bit less. By the time you get to the third streak, you should be able to see isolated colonies. So you're going to determine which isolated colonies might be Staphylococcus epidermidis and which are Staph marcescens or Serratia marcescens, because Staph epidermidis is white. It's a small white colony, and Serratia marcescens is usually a red colony, okay, if it's growing at 30 degrees Celsius. So we are actually going to incubate these plates at 30 degrees Celsius, which I just told you 37, but it's really 30. That's so that we can see the red pigment and the Serratia marcescens. They will both grow at 37, but they both will be white. So after we're done, we should see some white colonies. We'll pretend this one is white. And this one is red. Okay, that would mean we have a pure culture of Staphylococcus epidermidis, because this colony started from one single cell of Staphylococcus epidermidis. So it is a pure culture that can be created. So we would just need to take an inoculating loop using aseptic technique, take some of that culture, put it into a new nutrient broth, and we have a pure culture of Staph epidermidis. Same thing with the Serratia marcescens. If we pick the red colony, take an inoculating loop, okay, sterilize it using aseptic technique, take a little bit of that red colony, put it in the nutrient broth and grow it up, you will have a pure culture of Serratia marcescens. So we would have successfully taken a mixed culture and we would have separated so that we could get pure cultures of each of those two bacterial species. Okay, so that is street plate technique, and we'll go on to the next video, which will be selective and differential media.